Hello children, in the last class we have seen that in the previous class we have studied that how characters are transferred from parents to the progeny. Okay, gene is responsible for the transfer of character from parents to the progeny. Okay, now you know that gene is located on the chromosome. During the process of meiosis, the chromosome along with the gene segregate and it enter into the gamete. Okay, so the male gamete is having paternal characters and the female gametes are having maternal characters. Okay, when this gamete fuses and progenies are formed, progeny receives paternal and maternal character. So this is what you have studied in the previous chapter. In this chapter, we are going to study the molecular structure of a gene. So what is a gene? Gene is a segment of DNA. It is a segment of DNA. Segments mean this is the structure of a DNA. Okay. So what is the DNA? DNA is a nucleic acid. It is a nucleic acid. Nucleic acid are of two types, that is DNA and RNA. So what is DNA? DNA is deoxyribonucleic acid and RNA is ribonucleic acid. This nucleic acid is made up of three components. You can see here it is made up of nitrogen base, sugar and phosphate. Okay, nucleic acid is made up of nitrogen base, sugar and phosphate. Now we will see what are the components present in the nitrogen base. So in the nitrogen base is divided into two, that is purine and pyrimidine. Purine and pyrimidine. Purine are of two types, that is adenine and guanine, and pyrimidine are of three types. Pyrimidine are of three types, that is thymine, cytosine and uracil. Okay, these are the nitrogen bases. So after that we will see sugar. Sugar is made up of this sugar is a pentose sugar. What do you mean by pentose? Pentose means it is having 5 carbon atoms. So when you see the structure of a pentose, you can see like this. This is the structure of a pentose sugar. This is 1 carbon atom, 2nd carbon atom, 3rd carbon atom, 4th carbon atom, and this is the 5th carbon atom. So due to the presence of 5 carbon atom, this sugar molecule is known as pentose sugar. Pentose sugar is of two types, that is ribose and deoxyribose. Ribose and deoxyribose. So see, what is the difference between these two pentose sugar? They are exactly similar, but there is a slight difference. You see here the structure of the ribose sugar. It is having HOH in the first carbon. This is the 2nd carbon, HOH, 3rd carbon, HOH, and HCH2OH. This is the ribose sugar. Now we will see the deoxyribose sugar. Deoxyribose sugar is exactly similar to ribose sugar, but there is a slight difference. HOH is present in the first carbon atom. So here only you can see the difference. This is the second dash, that is second carbon atom. In the second carbon atom, oxygen is absent. Deoxy means oxygen is absent. Where? In the second carbon atom. Here you can see in the second carbon atom, oxygen is present in ribose. The remaining things are same. HCH2, we can write here as OH. Okay, so this is the difference between ribose sugar and deoxyribose sugar. The difference can be seen in the second carbon atom of the pentose sugar. In deoxyribose, oxygen is absent. In ribose, oxygen is present. Okay, now this sugar molecule, this is sugar, pentose sugar, is linked to a nitrogen base. This is the nitrogen base. Okay, this is sugar, pentose sugar. This pentose sugar is attached, that is the nitrogen base is attached at the first carbon atom of the pentose sugar by a bond called N-glycosidic bond. Okay, thus forming nucleoside. So what is a nucleoside? Nucleoside consists of sugar and nitrogen base. Sugar and nitrogen base forms a nucleoside. Now, in the fifth carbon atom of this sugar, this is the fifth carbon atom of pentose sugar. When in this sugar, in this carbon atom, when phosphate is attached, phosphate is linked by a bond called phosphoester bond, by a bond called phosphoester bond. This is bond, okay, and now this molecule here, oxygen, now this molecule is having nitrogen base, sugar and phosphate. Togetherly it is known as nucleotide. Nucleotide. So what is a nucleoside? Nucleoside means do not have phosphate. Phosphate is absent. That means it is having only nitrogen base and sugar. That forms a nucleoside. What is a nucleotide? Nucleotide consists of nitrogen base, sugar and phosphate. So this is one of the nucleotide. We'll see the next nucleotide. This is the second nucleotide. Okay, here nitrogen base attached to the first carbon atom. Here in the fifth carbon atom, phosphate is attached, okay, which is linked with the third carbon atom of the first nucleotide. Okay, and this bond is known as phosphodiester bond. It is known as phosphodiester bond, and this forms a dinucleotide. It forms a dinucleotide. Like this, you can see here in the fifth carbon, free phosphate moiety is there, and in the third carbon, you can see free hydroxyl group. So free hydroxyl group is present in the third carbon atom, and free phosphate moiety is present at the fifth carbon atom. So this nucleotide is in 5' to 3' direction. So when many nucleotides combine together in this form makes a polynucleotide chain. You can see in your screen the structure of the polynucleotide chain. The backbone of the polynucleotide chain is made up of sugar and phosphate. Nitrogen base is linked to the pentose sugar and project from the backbone. Is it clear? Now, we will see the difference between RNA and DNA. So, the first difference between RNA and DNA is in the sugar. As we know that in sugar, deoxyribose do not have oxygen in the second carbon atom, but ribose is having oxygen in the second carbon atom. This is the first difference between RNA and DNA. RNA sugar, ribose sugar is present in RNA and deoxyribose sugar is present in DNA. Second difference you can see in the nitrogen base. Nitrogen base. Here in RNA, adenine, guanine, cytosine and uracil is present. In DNA, adenine, guanine, cytosine and thymine is present. Instead of uracil, in DNA, thymine is present. Okay, so this is the difference between the second difference between RNA and DNA. Third difference is that RNA is having a single polynucleotide chain, but DNA is a double helical structure. So you can see here there is one more chain. Okay, this is the sugar molecule. So the first carbon atom is having nitrogen base here, and here you can see in the fifth carbon atom phosphate is attached. Okay, so the next sugar molecule this is nitrogen base. Here you can see the carbon, fifth carbon is attached with the phosphate. Now this is the 5' end, this is the 3' end. Here this is the 5' end. Why it is known as the 5' end? Because this is the fifth carbon atom. This is the 3' end, which is having hydroxyl group. 3' end. See the orientation of these two strands. Here in the first strand, at the top you can see free phosphate at the fifth carbon atom. Here at the top you can see hydroxyl group, that is in the third carbon atom. So this strand is having 5' to 3' polarity. That means 5' to 3' polarity. The next strand is having 3' to 5' polarity. These two strands are anti-parallel to each other. These two strands are anti-parallel due to the presence of, that means due to the orientation of the sugar molecule. See the orientation of the sugar molecule in these two strands. Okay, here the fifth carbon is at the top, here the third carbon is at the top. So that is why one strand is in 5' to 3' direction, and in the next strand you can see that it is in the polarity of 3' to 5' direction. Here you can see the nitrogen bases. These nitrogen bases are joined by hydrogen bond. Hydrogen bond. Okay, so in a polynucleotide chain, you have seen there are three types of bond. Glycosidic bond, which joins the nitrogen base with that of the sugar. Phosphoester bond, which joins the phosphate with that of the sugar molecule. Hydrogen bond, which joins the nitrogen bases. This DNA is acidic in nature. It is acidic in nature. Okay, now this acidic DNA is present inside the nucleus. It is present inside the nucleus. This DNA was first identified by a scientist called Friedrich Miescher in 1869. In 1869, Friedrich Miescher first discovered the identified the DNA, and he named it as nuclein. He named it as nuclein. Next, James Watson and Francis Crick. These are the two scientists. They made the model of DNA, model of double helical structure of DNA. This model of double helical structure is made based on the X-ray diffraction produced by Morris Wilkins and Rosalind Franklin. These two scientists took the X-ray diffraction model, diffraction, and on the basis of X-ray diffraction, James Watson and Francis Crick made the model of DNA. There is another scientist whose name is Erwin Chargaff, who proposed the base pairing. Base pairing means adenine, this is nitrogen bases adenine always pair with thymine and guanine always pair with cytosine. So between adenine and thymine there is 2 hydrogen bond, between guanine and cytosine there are 3 hydrogen bond. Now you can see this adenine and guanine are purine and this thymine and cytosine are pyrimidine. So what you have learned from here? Purine always pair with pyrimidine. So if you know the nitrogen base of one strand, suppose it is having A, adenine, it is having cytosine, you can predict the nitrogen bases of the next strand because adenine always base pair with thymine and vice versa, and cytosine always base pair with guanine and vice versa. So here they are having a triple bond, here they will be having a double hydrogen bond. Now this is the double helical structure of DNA. Now you can see in your screen the double helical structure of DNA. Backbone is made up of sugar and phosphate. 2 chains are anti-parallel polarity, that is in 5' to 3' direction and 3' to 5' direction. Bases of the 2 strands are paired by hydrogen bond, 2 hydrogen bonds between adenine and thymine and 3 hydrogen bond between guanine and cytosine. The next picture you can see here, it is a double helical structure of DNA. The 2 strands, you can see the 2 chains are coiled in right-handed fashion. The pitch of the helix is 3.4 nanometer, but it is written there as 34 angstrom. 34 angstrom means it is 3.4 nanometer, and there are 10 base pair in each turn. The distance between the 2 base pair is approximately 0.34 nanometer. Now we will study the central dogma. What is central dogma? The central dogma was proposed by Francis Crick. Okay. Central dogma of molecular biology state that genetic informations are transferred from DNA to RNA, that is mRNA, to protein. So this is the flow of genetic information. From DNA to mRNA is called transcription, and from mRNA to protein is known as translation. Okay. Now, here DNA can replicate into DNA. So that process is known as replication. Okay. Now, you can see that DNA transcribed to mRNA, mRNA is translated to protein. That is known as central dogma. But in some organism like viruses, especially in retrovirus, example of retrovirus is HIV. HIV is an example of retrovirus, where the genetic material is RNA. As their genetic material is RNA, they undergo reverse transcription. What is reverse transcription? That is, what is transcription? That is information transferred from DNA to mRNA. Reverse means from RNA, it is transferred into DNA, reverse. So that is the reverse transcription. And then from DNA, it is again transcribed into RNA, and then it is translated to protein. So here you can see reverse transcription. This type of reverse transcription you can see only in the retroviruses, which is having genetic material as RNA. Okay. Now, this is about the central dogma. Do you know the length of a DNA? We can calculate the length of a DNA if we know the base pair of the entire base pair. Human beings are having near about 6.6 into 10 to the power 6.6 into 10 to the power 9 base pair which is present in human being. Okay, we know the distance between two consecutive bases. What is the distance between two consecutive bases? That is 0.34 nanometer. If you convert this 0.34 nanometer into meter, you will get 0.34 into 10 to the power minus 9 meters. When you multiply this with the bases, total number of bases, we can get the total length of a DNA. So that is 6.6 into 10 to the power 9 into 0.34 into 10 to the power minus 9 meter per base pair. That gives an approximately 2.2 meter. That is, this is the length of the DNA. Just imagine if the length of the DNA in human being is 2.2 meter, where the nucleus size is much smaller, the dimension of the nucleus is much smaller. This much large DNA is packed inside the nucleus. So we will see now how this DNA is packed inside the nucleus. So our next topic is packaging of DNA. Packaging of DNA. First we will study about the prokaryotes. What do you mean by prokaryotes? Prokaryotes do not have membrane bound organelles. Okay, that means they do not have nuclear membrane. So when you see a prokaryote like bacteria, their genetic material is not floating in the cytoplasm. Actually, this DNA is negatively charged particle. The DNA are negatively charged. Okay, and inside this cytoplasm there is a protein component which is known as nucleoid, which is positive in charge. Okay, so this negatively charged DNA attaches with the positively charged nucleotide and forms the loop. So this type of package or arrangement you can see in the prokaryotes. So let us see what about the eukaryotes. In eukaryotes, actually they are having a nuclear membrane. Eukaryotes are having nuclear membrane. Okay, their packaging is somewhat complicated. Here there is a set of histone protein. There is a set of histone protein.
There is a set of histone protein. These histone proteins are basic protein which is positively charged, and these histone proteins are made up of basic amino acids like lysine and arginine. Lysine and arginine. These histone proteins are made up of basic amino acid like lysine and arginine. So it is positively, highly positive charge. Okay. Now DNA is negative in charge. Histone is made up of 8 molecules of histones. This histone protein is made up of 8 molecules of histone. So what are the molecules? There is H2A, H2B, H3 and H4 are the molecules of, these are the molecules of histone. These are the molecules of histone. That means H2A, 2 are present, here H2B, 2 are present, H3, 2 are present, H4, 2 are present. Total how many are there? Total 8 molecules are present. So this forms a histone octamer. Histone octamer. So octamer means 8 molecule. This histone octamer, around this histone octamer, the DNA chain wraps or coils around. Okay? And the free end of this DNA is attached with another histone which is known as H1. Thus forming a nucleosome. So what is a nucleosome? Nucleosome consists of histone octamer and 200 base pair of DNA. That makes one histone octamer. So many histone octamer joins together. Second histone octamer is joined by a DNA link. Now I'll show you in the screen how the two histones or nucleosomes are joined. Now you can see in your screen the 2 nucleosomes. These 2 nucleosomes are linked together by a linker DNA. Okay, see the 8 octamers forms the histone octamer around which a DNA coils and forms the nucleosome. Is it clear? Many nucleosomes, see many nucleosomes joined by a linker DNA to form the chromatin. This here what happens, some of the regions of the chromatin is loosely arranged. That type of chromatins are called euchromatin. Euchromatin. Some of the chromatins are tightly packed, that is known as heterochromatin. Okay, and this chromatin condenses to form chromosome. So in the form of chromosome, DNA is packed inside the nucleus. So when you see a euchromatin, that means where transcription takes place because it is loosely arranged. In heterochromatin there is no transcription. Is it clear? Students, I hope you might have understood the topic. In the next class, we'll study different experiment performed by various scientists to find out whether DNA is a genetic material or protein is a genetic material. Okay, thank you.